Hey, what going on, Ollie? We're going to do biology, unit 1, paper 2, right? That is the 2019 paper. So if you're here, welcome. Do we see how best we could pass these exams? Do we see how we could help out here? So question 1. Figure 1 shows a light micrograph of the transverse section through the primary stem of a dahlia plant, a typical dicot, right? So we could see what we see in here. We see is a light micrograph. We could see all kind of cellular details here. We could see 5 labels A, B, C, D, and E. So watch me here. One time they're gonna ask me what is what, right? So I'll just tell you right away. So A is the epidermis. You'll notice here that the epidermal layer, you'll see that it had two is like it's like kind of like a double line here. You want to make sure that when you're drawing this that you have that double line in there, right? So B is going to be this inner part right below the epidermis. So there's the cortex. C. So this whole thing here, you see that this is it kind of sparse looking. This is the pit or the medulla. Basically, just means the inside. So pit usually means inner layer, and cortex means outer layer, right? So this whole section, this whole thing here, so you're seeing like how you see a whole set of these here, like you see 1, 2, 3, 4, 5, and you see that there are varying sizes. Each thing here is called a vascular bundle. And what we have here is two parts of the vascular bundle, and you'll see that they're more pointing to, actually I'm too sure what they're pointing to here, but if they're pointing more to this inner part here, so this is the xylem, and this outer part here is the phloem. In the middle here is the cambium, but I think what they're really looking for here is xylem. So what the first thing that I want you to do here is in the space below construct a tissue diagram from the light micrograph of the plant section in figure 1. And this is 3 marks. So typically you don't necessarily need to label it. I would still say to label it, but it's not really for the marks because you can see the next question here is to provide annotations. You see I already have it typed up nice, so they wouldn't have to wait for me to type it out. So right, so this is what the diagram look like, and as it is not the best diagram in the world, the pencil point could be a little thinner, it could look a little neater, but as it is just a representation of what you want to draw. You would know what you're doing. You'll notice that there's no cellular details here. What you will also notice is that there's almost the same amount of vascular bundles here, but not exactly the same amount. But you don't need to put, as say, it's not a direct illustration, it's just a plan diagram where you just you just want to be representative of what you're seeing. You want to make sure that everything is proportionate. Proportionate being that you don't want to make your pit or your medulla small, at least compared to how the cortex is, right? You want to make sure that each one is appropriate size. You don't want to draw your vascular bundles chunky like ants, right? You want to make sure again that there's appropriate size, that they're proportionate and representative of what you see in the diagram before. You'll notice, remember I said the epidermis is like a double layer. You also want to make sure there's a double layer here that you put here. You want to make sure it as close to parallel or as close to concentric as possible wherever the word is. And yeah, so there you go, and you can see the labels here. Well, you fix up. So let me look at the next one. So they say provide annotations to describe each label structure A to E. And it's just 5 marks, so it's just 1 mark each, so it ought to be too detailed. Usually when they say annotation, they mean provide with a description or a basic description of the function, right? So the epidermis, as you see here, is the outermost layer of the dicot stem, and is usually covered with root hairs. Of course, root hairs is used for water absorption, right? And protects the underlying tissue. This is actually one of the main things that the epidermis is protection. And yeah, so again, either one could work. I just put the 2 here. Cortex, remember cortex means the outer layer. So the thing about the cortex is that it is usually used for mechanical support. And usually you'll find some amyloplasts in here, or amyloplasts, however you want to pronounce it. Those, they are basically starch storages. And remember, is a route we're talking about, and the route is a sink. That's usually where sucroses get delivered via the phloem, and you would get that being stored as starch there because, of course, the root there has no chlorophyll. It can't photosynthesize. Another thing we could note here is that the cortex usually differentiated into multiple layers, which is like the epidermis, the parenchyma, the collenchyma. You have an exocortical. Yeah, you don't need to say that here. I just put it to sound fancy. All right, so the pith is the inner part of the root, right? And basically, as what you can see here, so the pith here say is not dense. There's a lot of air spaces. If you remember how the spongy mesophyll was in the leaf, what that does is that it allows, you know, gaseous exchange. It allows passage of materials like solutions and nutrients. That's basically what the pith there for. And D and E is the xylem and the phloem. The xylem and the phloem, again, you don't have to put too much, is just the C-sex stuff. You have your xylem, which is basically dead empty cells. The walls lined with lignin, which is, you know, a very, very tough material, kind of like equivalent to cartilage, except cartilage are dead. And what it does is that it holds up the xylem. Now remember, the transport in the xylem is usually due to capillary action, right? It's usually due to root pressure and capillarity and transpirational pull and all them things, right? So what was happening here is that the flow through the xylem is unidirectional. Usually it's from the roots to the leaves, and it transports water and any dissolved ions in that water. All right, so now we have the phloem. The phloem, opposite to the xylem, is actually alive. It requires ATP, usually provided through these companion cells. Let's learn a little bit more that in Unit 2. And they are not made out of, you know, like dead cells like lignin and things like that, but, you know, made up of sub-tube elements. And key thing here is the function. The function is that it doesn't really transport water per se, but it transports photosynthetic products, basically synthesize organic food in the form of sucrose. And it does this from a source to a sink. So a source is usually a photosynthetic organ like a leaf, and that will make the sugars, and then it will be loaded into the phloem, which will be, you know, which will transport it to a sink, which is a non-photosynthetic organ like a root. And it basically like a delivery system, like a delivery truck, where the source would be the KFC and the sink would be a house. Come and give me some KFC. In this case here, give me some sucrose. All right, so let me look at the next one. The graph in figure 2 shows the change in the rate of uptake of two substances, substance A and B, as the concentration of each substance increases across the lipid bilayer. So if you look here, we could tell the difference right away. We could see A. Well, first of all, this is a straight line. Anything with a straight line, some letters is represented as a directly proportional relationship. Whereas B, you could see that is increasing, and then it sort of tapers off. It's kind of like the rate of diffusion. It decreases until what we have here. Well, we can see the rate of increase decreases until there's a plateau. Not that there's no diffusion occurring here, it's just that there's no further increase at B, and you can see that this is occurring at about 3. When it reaches about the concentration at 3, it stops increasing. Alright, so let me look here at the question. So they say describe the changes in the rate of uptake of substance A and B as the concentration of solute increases as depicted. Alright, so they don't want to talk anything about the types of diffusion yet. All they ask here is to describe the changes. You need to go onto the microscopic level with carrier proteins and they're asking that, right? They're just asking you to concentrate on this graph. Alright, so you see with A, there's a linear increase in the rate of uptake of substance as solutes in concentration increases. Easy. The more the concentration of solute increases, the more the rate of diffusion increases. That's depicted perfectly by the straight line. Alright, so B. The initial increase in the rate of uptake of initial increase in the rate of uptake as concentration of solute increases. So there's an initial increase. in the rate of uptake as this is increasing here, the solute, right? And this is quickly followed by a plateau or no further increase in concentration. It's just 2 marks, so you just need to put a basic description of what you're seeing there. Right, so the next question after this is asking here, what type of diffusion is each one? Right, so in this case here, A is simple diffusion and B is facilitated diffusion. Right, so now what they're asking for here is to compare the two types. So now we're talking, now we're doing we smart man work, right? So how are we going to compare these? So first, again, look at how much marks the thing is. And this is 4 marks. So typically, if you have about 2 of these points here, right, compare and contrast, you want to have at least 2 of these points here. You'll be able to get your 4 marks. I would say you could go for a little more because this is an easy topic. But let me go through it here. So we could compare and contrast, right? So you could have one comparison, one contrasting one. All right, so for both simple and facilitated diffusion, molecules and ions move down a concentration gradient. So this is true for either one of them. Whether or not is each one, if you have more on one side and less on the next side, you're gonna get it flowing from higher concentration to lower. For both simple and facilitated diffusion, no energy is required. So there's a passive process. It's unlike things like, you say, active transport, right? Both of them is due to the actual internal energy of the molecules. The kinetic energy of the molecules naturally moves them about. There's no additional energy needed from any cell or anything like that. So it's not like loading sucrose into a phloem, into the phloem. 3. Simple diffusion occurs through phospholipid bilayer, whereas facilitated diffusion occurs through the transport transmembrane proteins. Okay, so simple diffusion, usually the molecules are really small. So this would be like, things like carbon dioxide. They're able that's basically just a carbon and two oxygens, right? And that could pass through the lipid bilayer very easily. Yeah, again, because it's small, it's kind of like you have a little thing in the fence, and little small things could pass through that fence, right? And facilitated diffusion, however, is used to move bigger substances or usually polar substances. So for example, glucose. Glucose is 6 carbons, 6 oxygens, and 12 hydrogens. That's a sort of big molecule, right? And we could say, well, that can't really pass through the fence. That need to pass through a gate. It need to pass through a gate. And of course, if you have only so much gates, the gate's gonna get a little clog up from time to time, right? You're gonna get lines and things by the gate. And those gates there, those are carrier proteins or channel proteins. So facilitated diffusion requires the help of these transport proteins. Simple diffusion could just pass through the bilayer just so. Right, 4. Small and non-polar molecules move by simple diffusion. Larger and polar molecules move by facilitated diffusion. Same thing, the transport proteins help those larger ones to get across. The smaller ones could just move through the fence. Basically, they could move through the little gaps in the bilayer. Alright, so 5, facilitated diffusion may be initially faster than simple diffusion, but is plateau, basically what you see in the graph there. And it becomes saturated as solute concentration increases, thus the plateau. Simple diffusion is more of a linear relationship. The more concentrated the solute is, the higher the rate of diffusion going to be. Lastly, facilitated diffusion can be inhibited by substances or conditions which denature proteins. So it's kind of like if you have some things, if it's controlled by a gate, right? The gate in this case being a protein. If you have something that mash up the gate, like if you have a irregular pH or you have a very high temperature, and those things there can actually denature the proteins. They could change the structure of the proteins and they wouldn't function as they're supposed to. So simple diffusion is actually not inhibited by any of these things, because simple diffusion has no proteins involved, so it doesn't really matter. Simple diffusion, however, could be increased with things like temperature because temperature increases the kinetic energy of the molecules. All right, so you look at this one here. So they say arrange the following molecules according to their ability to diffuse through the lipid bilayer. You have carbon dioxide, glucose, water, and RNA. Begin with the molecule that diffuses the easiest. So the order that you want to go here is, you can think about the least complex to the most complex, right? And this is going to be the least complex here, going to be CO2. Again, it's a carbon and two oxygens. Yeah, water, H2O. What makes water a little more complex is that it has a dipole structure to it. There's a slight charge on the hydrogens and a slight charge on the oxygen, and that makes it a little more complex. Glucose is C6H12O6, is a lot larger than the other two. And RNA is quite complex in itself, right? If you do anything in modules, if you remember anything in that, you'll see how complex that is. It's also the largest of these four. So basically, that's you just try to justify why you have that order. So the correct order is probably about 2 marks, and each one of these is 1. So CO2 is small, is non-polar, and is lipid soluble. It can actually pass through the bilayer. Water is small and polar, so usually it cannot really pass through the bilayer because because of its dipole structure, also because the tails of the bilayer are hydrophobic, so you wouldn't really expect too much water to pass through there. So usually these would transport through special proteins called aquaporins. Glucose is large and polar, and these of course require these gates in order to pass through transmembrane proteins. And RNA is very large, it's the largest of all. It's highly charged, very polar, so these here, typically like the RNA that would pass through would be like messenger RNA, which has to that has to kind of carry the instructions to the ribosomes to build proteins. And those typically also have to pass through transmembrane proteins, right? They're very large compared to the others. All right, so this next one here, there's a question that pops up from time to time, and they say, with the aid of an annotated diagram, explain the organization of membrane lipids. Discuss the structure of a cell membrane, specifying the organization of membrane lipids and the various ways in which proteins can associate with the membrane. And the drawing is, so the way to explain to us is not that the drawing, there's a big question here. And this is 8 marks, right? So your drawing here might be like just 2 marks because it's not a hard drawing at all. And you want to put as much points as possible. So first, let me show you what the drawing would look like. So we can see here. So here is the outside of the cell, and this is the inside. This is the cytosol of the cell. This would be stuff that comes in. So if you have a piece of RNA, mRNA, it will be coming from outside all the way through here. And you can see the lipid structure here. So these are the phospholipids. They have a phosphate head, and they have fatty acid tails. And this phosphate head here, this is hydrophilic, which means it likes water. And these fatty acid tails are hydrophobic, which means that they don't like water. And this is basically what forms this double membrane structure here. This is what keeps them, this is what keeps them in this order. And so what you're gonna see throughout is that these sort of turquoise structures here and these lilac structures, these are proteins. And these proteins here, these are going to be, they could occur throughout, which means that they span the entire length of the membrane, right? And these here are going to be integral proteins. So these integral proteins here, the ones I see in like this one and this one and this one, these could act as channels or act as carrier proteins, which means that they could take stuff from the outside and send it inside, or vice versa. These other proteins that you're seeing here, these are peripheral proteins, or extrinsic, right? These integral ones are also called intrinsic. And these usually just linger on the outside, and what would usually be attached to them, always not shown here, are receptors for things glycoproteins. What would usually be attached to them are receptors for cell signaling and things like that. Next, they have cholesterol. Cholesterol is usually found between the phospholipids, and the cholesterol sort of helps maintain the stability of the layer. They respond to temperature, and that response to temperature helps to either, you could think of it as spacing out the phospholipids or bringing them closer together. So this is typically what you draw. Now, as for the points you want to make, I put 8 points here, but you don't need to make all 8. What you want to have here is at least 6 keys, I would say, and you want to be able to describe everything that I just talked about, right? So talk about all the different parts. It's kind of like you're having the annotations. So I'll just read some of them. In aqueous environments, this should be the hydrophilic or the water-loving part of the phospholipid or the polar end. The head will interact with the water and face outwards into the aqueous environment, inside and outside the cell. So as you see, we had the heads, and the heads was sort of facing outward, and the tails was facing inward because those are hydrophobic. Those hiding from the cytosol and all the watery substances there. So you can make this. The hydrophobic, water-hating or non-polar end, the tail, will stay shielded from the water on the inside, right? They hate water, they hide it. Glycolipids and glycoproteins help stabilize the membrane structure by forming hydrogen bonds with water molecules outside the membrane. These are used for cell recognition as well, receptors, cell signaling. These are like little antennae that you'll see coming out of the protein structures. Most proteins float about in the phospholipid bilayer, forming a fluid mosaic pattern. Right, so it's kind of like the way on which to describe it is imagine the protein structures as icebergs floating in a phospholipid ocean. So imagine you have an ocean full of phospholipids. The proteins are like icebergs sort of flowing there. They're not static, right? They're actually moving about. We call it the fluid mosaic pattern or model. Some proteins penetrate only part of the way into the membrane, while others penetrate all the way through. And the ones that I say would act as transport for transmembrane proteins. They would take stuff in and take stuff out. They're like gates. Proteins can be found at the outer inner surface of the phospholipid bilayer. These are the peripheral ones. Peripheral, they're kind of just on the outside. They're not penetrating all the way through. They're loosely bound, and these could serve as points that glycolipids and glycoproteins could attach, again, for cell signaling. Cholesterol helps to maintain the fluidity of the membrane as temperatures change. It prevents the membrane from becoming too rigid, firm, or stiff. Of course, if it becomes too firm or stiff, you're gonna have that's gonna slow down your simple diffusion, your gaseous exchange, and things like that. And that's bad, right? It also will hinder the movement of the proteins in the membrane. Remember, I said the proteins are like icebergs floating in a phospholipid ocean. So any 6 of these, once you put a basic description of what you see is that's what it is. Alright, let me look at question 2. Table 1 shows the relative growth rate data for sensitive strains and resistant strains of S. typhimurium bacteria exposed to varying concentrations of tetracycline antibiotic. And what you can see from the table here is that you have an increasing concentration of tetracycline, which is an antibiotic, and this is in micrograms per milliliter. And you can see that there are two strains of bacteria. There's the sensitive strain and there's the resistant strain. And we have the growth rates of those. Now, of course, the sensitive strain means that it's more vulnerable to the antibiotic, and the resistant strain means that it's less vulnerable, right? It sort of it was sort of be able to is able to overcome the antibiotic. So if you look at the basic thing that you're seeing here is that as the concentration of tetracycline went up here, you can see it went up to 0.8. You'll see here that the sensitive strain is the one you expect dead. So this is actually going down as this rate the more and more antibiotic being added, right? Because again, it's sensitive and not as strong. Now, what you notice here about the resistant strain, as this tetracycline concentration going up, this more or less remaining the same. It remaining sort of constant, at least within a within a margin of error. And the question does look harder, but sometimes you just have to use your common sense a little bit. Just look at the numbers. You take easy. So the first thing here, they say on the grid provided on page 11, plot a line graph showing the relative growth rate. There's the y-axis versus tetracycline consider x-axis. Well, they don't give you the thing. You just have to draw it. 6 marks. That's an easy 6 marks. So it was plotted here on Excel because I'm too lazy to draw the graph, and let me see what was being shown here. And you'll see here the relative growth rate, the sensitive strain, the resistant strain. If you're doing this in a graph page, of course you can't have colors, so one would be little dots with circles, and the next one could be X's just to differentiate. Don't forget to put your your key or your legend, and don't forget to put a title. May I put on here? And you'll see here the sensitive strain, which is blue, while as the antibiotic concentration going up. You can see this going right down. Whereas the resistant strain, well, he good. He sort of little fluctuations here, but remaining almost the same. So this is what your graph would look like, right? You would know when you draw it. So you look at the questions that I have here now. So they say outline the effect of each of the following antibiotic concentrations on both bacterial populations at 0.1 microgram per milliliter and 0.7. Okay, so there's the one to know for each one. So 0.1, let me take a look at it. I'll put it on the side there. And we could see here that the resistant strain is apparently unaffected. It has a slight increase or slight fluctuations by this increase in antibiotic. And the relative growth rate is, from what you're seeing on the graph there, is close to 1. By the end of the thing, it kind of hovering around 1.01. So when they say, outline, try and put some numbers in there, see like what is the growth rate about, what it end up as, things like that. So there's about 0.1, right? The concentration of 0.1 microgram per milliliter. So that's resistant. So the sensitive one. So the sensitive one you see is that the rate is greatly reduced. The more antibiotic you add in is the more it drop in. And at the end of this, it dropped to 0.67. And initially the thing was 1, and just adding 0.1 microgram per milliliter, it dropped to 2/3 this amount. Alright, so 0.7, the growth rate is sort of the same. It's still unaffected because it's resistant. Relative growth rate is close to 1. Same thing. And at 0.7 now, this is where we take a drastic drop. At this point here, plenty of the bacteria dead in the sensitive strain. So the growth rate of the sensitive strain is greatly, considerably, or severely reduced or decreased. Note here, you just don't want to say decreased, right? You want to say greatly decreased because the thing dropped from 1 to 0.08, right? There's a big drop, right? The growth rate reduced to 0.08. Again, you want to put some figures in there. The thing is 4 marks. Show that you're able to read a graph. Alright, so look at question 3. Explain how the differences between the growth rates of the bacteria could lead to natural selection. Well, 4 marks, you're gonna have about 4 of these points here. Well, it's easy. You don't kill something, you're gonna make it stronger. That's natural selection, right? Survival of the fittest. The thing gonna become stronger and it's gonna become familiar with the antibiotic, and eventually you're gonna have a problem. It have a bacteria called MRSA, methicillin resistant Staphylococcus aureus. You can look it up. And that's a major problem in hospitals, because hospitals they have antibiotics there all the time. And yeah, that bacteria sort of become resistant to a lot of antibiotics. So it's a problem when you go in the hospital and you get that there, you're in trouble. So explain this and link it to natural selection. So bacterial strains differ in susceptibility to the antibiotic. Lower growth rates in sensitive bacteria strain on exposure to the antibiotic, and there are higher growth rates in the resistant strain. So you know the difference there, right? So that's a mark. Now, when exposed to the antibiotic, higher growth rate promotes survival in the resistant strain. Lower growth rate reduces survival in the sensitive strain. So far, notice that in the sensitive strain, not all there, right? You still had 0.08 or whatever growth rate by the end. Growth and reproduction continues in the resistant strain on exposure to the antibiotic. Again, you're killing the thing, you end up making it stronger. Growth and reproduction reduced in the sensitive strain. The antibiotic acts as an environmental or a natural selective force, so it acts as what we call a selective pressure. What this thing is doing is that is introducing it to a factor that it has to be able to overcome. Of course, the weak ones are going to die and the stronger ones are going to survive. So the ones that survive, those was the strongest of that population. Those they're eventually going to reproduce, and the overall population is going to be stronger. The surviving members of the sensitive strain was exposed to high levels of tetracycline can eventually proliferate or reproduce. Again, they try to kill the thing, not all of it gonna die. The few that survive, those are the strongest, and those gonna reproduce, right? Proliferate. And they're gonna make back that whole population. The whole population gonna end up stronger. The surviving members of the sensitive strain can thus become a population of a new resistant strain. Right? So just to sum it up here, you try to kill the thing, most of it dead, not all of it dead. The ones that are dead, them staying behind, they're starting to reproduce, they're starting to proliferate, and them few strong become plenty strong. And thus the cycle is continuing like that. That's natural selection. You look at the next question. Alright, so the figure 3 below illustrates how mutation brings about genetic variation. And here we have a normal protein, and we could see that the DNA here is GAG. But we have a mutant protein, we have a point mutation or substitution. Instead of a adenine here, we have a thymine, right? A T. So the one to know now, they say eventually what's happening here is that the complementary pair of this DNA, this is of course going to be different. And then this is the RNA is going to translate, sorry, the RNA is going to be built differently. And of course, that is going to translate into a whole different protein, right? And we can see here that the protein GLU is being built to be the normal thing. But Val, right, or valine is going to be the mutant protein. So something is kind of like you have a blueprint for instructions, and somebody change a word in the blueprint, and all of a sudden the whole build looking different in the end. The DNA is like the blueprint. The RNA is like who translates in the blueprint. It's kind of like who going to build the thing, and the protein is the building. So if the blueprint wrong, the whole building might come out wrong. So they're saying here completely missing codons. So we know a good way to remember what is what here. So we know that A is paired with T and G is paired with C. Well, this is for DNA. And for RNA, A is paired with U. uracil. So I used to remember it as apple in the tree and car in the garage. Apple in the tree, car in the garage. But for RNA, it's apple under the tree, like the apple fall. Apple in the tree, car in the garage. So if you are car in the garage, well, we know that this G is going to pair up with a C. We know that this T is going to pair up with an A because apple in the tree. And the G here is going to pair up with a C. So this first thing here is going to be CAC as what you're seeing here. So the RNA version of this, right? So the car is going to be in the garage. The apple is going in the tree, right? But it says it's not in the tree, it's under the tree because the RNA has uracil, not thymine. And car in the garage. So the RNA version of this is going to be GUG. And you can see here that this translated to become GAG, the normal thing. The mutant one is GUG, and that is building a whole different protein. Right, so CAC is your first one, GUG is your second one. So they say now, with reference to the mutation in figure 3, explain how mutation brings about genetic variation at 3 levels, right? The DNA level, the protein level, and the cellular level. Okay, so this is, and let me see how much marks this is. This is 6 marks, right? And so you're gonna have, I would say, the number of points could differ, but I would say the DNA level have about at least 1 point, protein level at least 2 or 3, about 3 points, cellular level 2 points, basically what I have here. So the DNA level, what happened here? Say you had a point mutation, you had one letter, one nucleotide changed to something else, right? And in this case here, you had instead of a A, you had a T. So that gets the messenger RNA turn out different. The complementary DNA turn out different. Messenger RNA turn out different. And thus it was built differently. It's translated and built differently by the ribosomes. So what's happening here? So we had a single point mutation or substitution, a nucleotide being replaced by another. So when you have transcription, that's the building of the mRNA molecule, which is reading it and building it. And that would have been different as well, wouldn't be the same thing. And like again, the blueprint change. So that's at the DNA level, the lowest level. So at the protein level now, where you're building this, you're making the building now, the protein is like the building. So it's going to code for a whole different amino acid, valine, right? Instead of this, the normal one, GLU. The protein primary, so remember, proteins have, you can think of it as 4 structures: primary, secondary, tertiary, and quaternary. So the primary structure, which is just the polypeptide chain, the chain I make no twisty, twirly helix or nothing yet, right? But just the chain itself would be affected. And the sequence of amino acids would be affected, right? And as a result now, this would result in different shapes, different secondary and different tertiary structures. So what's happening here now is that the fold, and when they fold, the kind of bonds, there's disulfide or hydrophobic interactions or hydrogen bonds, all this is going to be different because the blueprint's different. So the entire thing is going to become distorted. The protein is not going to have the same shape. All of this is going to get affected. In the end now, not just the shape would be affected, but the function would be affected. It's kind of like if you have a machine and this machine is supposed to be a square, but instead it's a circle, the function is going to change. So at the cellular level now, really what they're talking about here with this example, with this one mutation is sickle cell anemia. Sickle cell anemia is a hereditary disease, usually caused by a point mutation. Yeah, one little thing coding for a different protein in the red blood cell. What happens there is that usually what happens is that the red blood cell is supposed to be able to have this a biconcave structure. Instead, what they end up having is this sort of crescent moon sickle structure. And as a result, it's because of the abnormal hemoglobin, it doesn't hold oxygen as efficiently, and thus that person with sickle cell becomes fatigued. So at the cellular level, what's going to happen? So the cell structure, if the protein structure is gonna change, the cell structure is gonna change. So the cell structure can be adversely affected. So you see, it's from the ground up. You do the thing wrong in the beginning, you're gonna echo and go up, and you're gonna get worse and worse and worse. So in this case here, abnormal hemoglobin causes red blood cells to have a sickle shape. You don't need to put that there. This is just but that's the example that I give there. So if the structure of the cell change, again the function of the thing gonna be affected. Same thing with the protein function. So the example with sickle cell here is that the red blood cells, they don't have the longevity, so they die prematurely. This could lead to anemia. The blood cells are inflexible. They can't really um flow, they can't have this nice flow through the capillaries. They get stuck in the blood vessels. It could cause hemorrhaging. It could cause less oxygen being able to be transported, right? Asphyxiation and so on. As a overall, they also have lower affinity to oxygen. They're less likely to bind oxygen to them, basically. So once you could explain on the basic level, you don't have to put the examples as I put here. Here by 6 marks. Right, so the last thing we have here is an easy one, is just to discuss the process of natural selection. And there's the amount of marks. The marks is below the sketch here. So this is 8 marks. So you see I put 9 points, right? So the first point here, variation occurs between individuals in a population. Variation due to mutation, crossing, or sex reproduction. What does that mean? Basically, mean everybody does look different, everybody different in little ways. And as a result, you're gonna have a larger gene pool. Some people have blue eyes, some people have brown eyes, some birds have thick beaks, some birds have thin beaks, long beaks, and so on. Now, this variation here is good because the variation allows for um, not just diversity of genes, also diversity of lifestyles. So, for example, now the if birds have thick beaks, what would happen is that they could eat nuts instead. They could eat seeds. If they had thinner beaks, they could instead feed on worms or insects. So we learned about this with Charles Darwin already, right? And as a result, they have less competition among individuals. So with this variation here at one, each generation produces more offspring than the environment can support, right? So this is the next one of Darwin's like little things that he postulated. There's a tendency for overpopulation. You have a dog, the dog gonna make 6 puppies. They're not gonna make one because that one might die. It makes six. Three of them might die, three of them might survive. You have a seahorse, the male one is get pregnant, and there's let out 2,000 little ones. The 2,000 little ones not gonna survive. In fact, it probably just gonna 20 of them survive it, if that. You get a leatherback turtle are gonna lay 100 eggs, only two of them might make it into the ocean. So all of that there promotes survival. The population increases, but resources are finite, so you end up with competition among the individuals. The environment applies selection pressures to select the fittest. Selection pressures could be food, it could be a predator, it could be a disease. Basically, it's something that the organism had to overcome. If it have a lack of food, but somehow they could get food. If it have a predator, but somehow they could avoid that predator, they could camouflage, they could spray some kind of bad smell, bad spines, all those things there help them to gain fitness in the environment. Individuals that are better adapted or have advantageous genes, alleles, or traits survive. So again, as I said, the ones that could camouflage, the ones that could fit in, the ones that able to find food that the others can't, the ones that are able to store food better, those are the ones that survive, the ones that are able to overcome diseases better than others. So the individuals that survive reproduce or transmit these advantageous genes or alleles. So for example, if one is a particular color and it could survive in this dark forest or this forest that has soot all over the wood, like the peppered mutt, they're able to pass down those genes and they're able to survive. and pass that on, so the offspring also able to survive. And as a result, this will continue once those pressures are still there, because they'll be able to overcome those pressures. Individuals with the non-advantageous genes or alleles do not get to pass them on to the offspring and thus do not survive. If you have um, a place where only black mutts would survive and you have a white mutt come, and the white would be more easily spotted by the birds and would get eaten. So that's a non-advantageous gene or allele, right? They wouldn't survive. They might be advantageous in a different environment, but probably not in this forest filled with soot. Frequency of the favorable beneficial genes or alleles increases in population over time. So as a result, now a lot of these ones with the beneficial genes, they're gonna um, reproduce a lot, they're gonna proliferate, and they're gonna increase their population until another pressure shows up. So after many generations, the population evolves, right? Or the population becomes better adapted to this to its environment. So the same thing with when you had Darwin's finches, you had some of them who had thin beaks, some of them who had thick beaks, some of them fed on seeds, some of them fed on insects, and thus as a result they didn't need to have competition, and they was able to separate into different niches and was able to survive like that. And that's how that worked out. Here we go, Module 3, Reproductive Biology, Question 3. Alright, so gametogenesis is the development and production of the male and female germ cells required to form a new individual. Right, so remember, gamete is a sex cell. Basically, they're talking about sperm cells or oocytes, secondary oocytes. So they say draw a well-labeled flow diagram of the process of oogenesis starting from the primary oocyte. Okay, so this is a kind of dreaded question because this is the part of reproductive biology that is getting a little confuffling, if you know what I mean. So let me go through it here. I have a nice diagram to show you. Alright, we'll check what's going on here. So don't get frightened. When you see this here, I try to break this down as easy as we could. So you see that we start with a primary oocyte. A primary oocyte is, you could think of that as when the girl is born, the germ cells inside the ovary, which is called oogonia, those they eventually differentiate, well, they eventually become primary oocytes, right? Now, this development continues after birth, and you'll see here that these are diploid, right? They have the full set of chromosomes. There's not any haploid cells, there's not any gametes or anything yet, right? They're just in the ovary. Now, what happens here is that you'll notice that meiosis 1 is that it stops at a certain point. It'd be like, what? Well, why does it stop? The thing is, the thing starts, but the primary oocytes don't actually become secondary oocytes until after puberty. So, after puberty, what happens is that the girl will start to ovulate and she would start to release, well, what we would call eggs from the ovary. The thing is not eggs, right? The thing is secondary oocytes, but I don't know why in Form 5 they used to call it eggs. The real name for the gamete is secondary oocyte. They become eggs eventually, but when they actually come out of the ovary, they're called secondary oocytes. They probably feel we was too stupid to say secondary oocyte, but that's what it is. So we had this primary oocyte that's a diploid, has the full 46 chromosomes, and only after puberty the meiosis finish and you start to get haploid cells instead, right? But you notice here that we have a next thing called a polar body. So after this primary oocyte makes two daughter cells, secondary oocyte and polar body, you'll notice here that they're not the same size. They don't have the same distribution as cytoplasm. Secondary oocyte have much more than the polar body. Well, remember, a gamete have to have 23 chromosomes because this oocyte had a mix-up with a sperm that have an extra 23 chromosomes. That would make a zygote that have 46. So this polar body is just a place to throw away the other 23 chromosomes. It's just a dustbin. It's just a genetic dustbin. Everything, this would never form a baby. It is not a gamete. This cannot be fertilized. In fact, there's two things that could happen to this. It could either form two other polar bodies, undergo meiosis again, meiosis 2, and form two other polar bodies. Or this thing might just break down and degenerate. It really don't matter. The thing just going to, it don't matter in the end because it doesn't form anything. It's just a place to throw away the other 23 chromosomes. So the secondary oocyte is what you would think was an egg back then. This is the thing that gets let out during the ovary during ovulation. Now, what happens here is that once one of these forms, you notice here that there's a next stop phase. And you'd be like, well, when does this become an egg? Well, what happens is that when the sperm fuses with the secondary oocyte, right before it becomes a zygote, right? What happens there is that it becomes an ovum or an egg just for a little while before the DNA starts to combine for it to become diploid. But the secondary oocyte, only after a sperm cell hits up with it. That's when it finishes meiosis 2 and becomes and splits to become an egg and a second polar body. The second polar body just degenerates. Nothing happens. It cannot get utilized. It cannot form a human. It can't even form a mutant human. The ovum or the egg is what will eventually become the zygote, and that there will, that is what will become the fetus and the baby eventually. So just to wrap it up real fast, you have a primary oocyte. These are formed basically before the baby is born, right? Before the girl is born. These are diploid. This splits. One becomes a polar body. This polar body may either degenerate or continue meiosis and form two polar bodies. These here, none of these things here are viable. They basically are just dustbins to throw away the extra 23 chromosomes so that the thing could be haploid. The other one that's found that contains a lot more nutrients, a lot more cytoplasm is called a secondary oocyte. The secondary oocyte is haploid, and this is what is formed once a month and what gets ovulated. Now, the process of meiosis does not complete. What happens is that the process of meiosis pauses until the secondary oocyte fuses with a sperm cell. And when it fuses with a sperm cell, it doesn't immediately become a zygote. What happens is that it finishes meiosis, like, hey, it kind of like it forget to finish meiosis, and now I need to hurry up and finish. It finishes meiosis, and this splits into two daughter cells, which one would be an egg or an ovum, and this is also haploid, and the second polar body which degenerates. The egg or the ovum, which is haploid, right, that will fuse with the DNA of the sperm, which is also haploid, to become a diploid zygote eventually. But that's not part of this question, right? And I know it could be a confusing thing because obviously it's not very straightforward. But once you focus on here and we just remember that there's a place we need to put the extra 23 chromosomes, all should be good and righteous. And in the end, you'll see that you end up with 4 daughter cells being formed, which is what is characteristic of meiosis. All right, so look at the next question here. State the role of one named hormone in oogenesis. Well, I put 6 here. Here, to just name the hormone as 1 mark and state the role as 1 mark. And it have plenty to choose from. You all could just read and see. I would say that easy one is FSH. FSH to stimulate the growth of the Graafian follicles in the ovary. That's basically the cells that would become oocytes. Eventually, it stimulates the development of the primary oocyte within the follicle to form the secondary oocyte. That's a good one. And next one is the the LH, luteinizing hormone, induces the rupture of the mature Graafian follicle and releases the secondary oocyte from the, from the ovary, basically causes ovulation. All right, figure 4 shows a diagram of a sperm cell. You could see 4 parts labeled A, B, C, and D. So A is the head, B is what you call the neck of the sperm, C is the body or the middle piece, and D is the, well, the tail or the flagellum. And the next part of this question here is asking it to provide an annotation on one function of each of the structures labeled A to D. So we have plenty thing. We just need one from each thing here. So we'll just go through each one. So the head, the head of the sperm contains the nucleus. The nucleus contains the DNA. And we are 23, well, as a human, you have 23 chromosomes, a haploid set. It also, so how the sperm, how it would actually penetrate the egg, right? You could think of it like it has had to digest its way through. It don't just poke and prod, it needs to actually use an enzyme. So the head also contains an area called the acrosome. It contains hydrolytic enzymes. These enzymes would help it actually get its way into the egg, right? Through the layers that you call the zona pellucida, right? So it could reach the zona pellucida. So it allows for the attachment to the zona pellucida and fuses with microvilli of the secondary oocyte membrane. So the head also has like these little attachment points that will help with the fusion of it. The neck. So the neck now, what that does is, well, the obvious one is that it connects the head to the tail. The head to the tail and the middle piece, right? But what happens here is that the neck contains centrioles, and these centrioles here, these help with differentiation of the sperm cell to actually form the flagellum, the tail. The middle piece is, you can think of it as the body of the sperm cell. So this is actually where you'd find most of your mitochondria. The mitochondria, as you know, is to produce ATP, right? And what happens here is that the middle piece is what's provide the actual energy that allows for the movement, the wave-like propulsion of the sperm, so it could actually be able to swim through the uterus, through the fallopian tubes until it meets the secondary oocyte for fertilization to occur. So the middle piece is, you can think of it as like the engine, that allowing it to move. The tail, well, everybody know about the tail. The tail of the sperm, probably the thing that show up the most when you think about it. Well, these move in a kind of lashing, whipping motion. The term flagellum is actually Latin for whipping, and that allows the sperm to swim in the fluid medium, which is the semen. So it allows it to be motile. It allows motility. It's able to move by itself. And just one mark for each one, so you just need to pick one for each one. So this next question here, I really sure what they're asking for. Does it relate any one structural feature of a sperm cell to its functions? So to me, that's what we just say. But if we think about, well, what is the function of the sperm cell? Well, is to actually reach the egg cell and fertilize it. So I would think one of the things to say is that, well, the shape of it, the streamlined shape, the flattened head, the elongated tail, cylindrical body, all of that increases its ability to swim. So it can actually fertilize the egg. And I guess we should I should put out there, it facilitates movement towards the secondary oocyte. That's really all I could do, because I mean we already say the things before, so I'm not really sure what they're asking for there, but that's my educated guess. Alright, so this next one here, a little tricky, but not too bad. Self-incompatibility is determined by multiple alleles at the S locus. Complete the following table using the key given, right? So a minus means no fertilization is possible, a plus means fertilization is possible, a minus and a plus means only half. So here we have S1, S2 is the genotype of the male parent. And basically what it says is that it cannot fertilize, it cannot, it cannot fertilize those of the same genotype. What this is doing is that it is trying to allow for diversity. So if it has the same S1, S2, it's not going to be able to fertilize it. So this one here, you see I have a minus. This is because it has 1 and it has 2. Now this one has S1, S3. So it has 1. So any of those pollen grains that have the S1 allele would not be fertilized. But S3 is not, is not present in the male parent. So any S3 ones will be able to fertilize. So the S1 ones will be minus, that means no. And the S3 ones will be, yeah, that's the plus. And S3, S4, but since these don't have any S1 alleles and S2 alleles, all of these would be able to be fertilized. So what this is trying to do, as I said, is it is trying to reduce the amount of offspring that would just have S1 and S2. Is trying to create diversity. So that's why it's called self-incompatibility. It's trying to prevent, let me say, like self-pollination, for example, right? That's a bad example, but it's trying to prevent, it's trying to say opposites attract, if you know what I mean. It's trying to say, well, you real different from me, so are we real differently, we get together. That's basically what it's saying. You see this S1, S2, and this, you see, nah, we too similar, we can never be together. Oh, you know the thing. Alright, so here we have some big questions. And I put a lot more here than you really need to put, but let me go through it one by one. Differentiate between each of the following pairs of terms. So we have incompatibility and male sterility. They might sound similar, but both of them different. And so let me look at each one. Self-incompatibility, I just say, if a pollen grain has an allele that is the same as one on the stigma, it will not germinate, or the pollen tube will not grow all the way through the style. It's self-incompatible, right? So it's an inability of a plant with functional pollen to set seeds when self-pollinated. It needs to get pollinated by something else that don't have the same alleles, right? So male sterility, on the other hand, means that in terms of plants, the pollen is just not there, or the pollen is non-functional. It's not able to fertilize anything. It's like with a man who's shooting blanks, right? He still have sperm cells. They're just, they're just inactive, or they don't work good. So it's the failure of a plant to produce viable pollen grains. Plants that have the mutant allele cannot self-pollinate, and they have to be cross-pollinated. So whereas they might have female sex organs, the male ones just don't work, so they have to be cross-pollinated. The one we have next is monoicy and diicy. Monoicy, just to give you a brief thing here, mono means one, di means two. Monoicy actually means one house. Diicy means two houses. So what happens here is that monoicy means that, imagine you have boys and girls in the same house. So a plant that exhibits monoicy is those that have both sexes. So they could have both the male and the female organs on them, or they could have male and female flowers, but on the same plant, right? You have boys and girls in the same house, right? Monoicy means one house. So plants that exhibit monoicy have unisexual reproductive organs or flowers, and I say it have both sexes on the same plant. Now diicy, which means two houses, two houses. A dioecious plant, however, what this would be is like you have a boys' school and a girls' school. You have all male plants and all female plants, right? Male flowers, female flowers, like cannabis, for example. And a dioecious plant, right, has separate sexes with individuals producing either male or female. So the male would be on one plant, the female in the next plant, two houses, boys in one house, girls in the next house. So the next thing we have to compare here is protandry and protogeny. So pro, just to break down the word here, pro means before, right? Before. And andry, right? Or andro means male, gyne means female. So if you think about it, protandry means male is formed before. Protogeny means that female is formed before the male. So protandry basically means that the male parts, the stamens, anthers, and androcium, develop ahead of the female parts, the pistil, the stigma, the gynosium. Or the pollen grains would mature and are shed before the pistils are ready to accept them. What this basically does is that it prevents self-pollination from occurring. It's kind of like, well, the man wake up, but the girl's still sleeping. And so thus nothing could happen, if you know what I mean. The man reach early, the girl come in later. Protogeny is just the opposite. Is the female wake up, the man still sleeping. So in this case here, the pistils, the stigma, the gynosium mature and accept pollen ahead of the stamens, the anthers, and the androcium, before those could even develop. So what happens is that, again, it's because both of them are active at two different times or two different parts of the year or two different stages of development, they would never be able to self-pollinate. They would always need to cross-pollinate. And again, this ensures the widening of the gene pool and diversity. Right. I believe this the last one. I thought two more. So the next question here is explain two major genetic consequences of each of the following methods of pollination. So we have self-pollination and we have cross-pollination, and this is 8 marks, and there's the final question. So what we'd want to do, I have 4 here for each one, but you just pick 2 and you know elaborate on it. So two major genetic consequences. So this could be positive or negative. Self-pollination is not the same as asexual reproduction, but it has some of the tenets of it, which basically means that you have you have less vigor, you have a narrow gene pool as a result. So self-pollination it promotes inbreeding. There's less genetic diversity than cross-pollination, because cross-pollination you would have plants with other environmental and selective pressures. You'd have plants with other advantageous or disadvantageous alleles. You know, you have a lot of different things mixing when you have cross-pollination. Self-pollination is just, you know, you just day in the house, right? Everybody day in the same house. So there's a greater chance of a loss of alleles through inbreeding compared to cross-pollination. So self-pollination could work better if you're trying to preserve certain alleles. Like if you have, for example, a flower that has a very bright flower, like a red rose, and you want that to carry on through the offspring, then yeah, you would self-pollinate if you want to sell them roses and things. So two, variation and hence adaptability to change environment are reduced. There's a restriction of the gene pool, restriction of evolution. So self-pollination, again, you have less diversity. So if a disease was to hit some of these flowers, they would affect much more than if they were self-pollinated, than if they was cross, because they might not have the genetic diversity to overcome a disease or a pest. 3. The vigor decreases with prolonged self-pollination, which increases susceptibility to pests and diseases or decreased yields. Same thing as above, you have less diversity. If a pest could feed on one flower, it might as well feed on all. But if it only could feed on a few, at least some would survive. And 4, it preserves genetic makeup of well-adapted plants in stable or unchanging environments. This is like in a greenhouse, for example, somewhere that is run by humans or stabilized in some other way. So if a given genotype is well suited for environment, self-pollination helps to keep that trait stable. So you won't have any quote-unquote pollution of the genotype from outside. In this case, here is usually artificially controlled. Like if you want to sell flowers of a certain color, you would try to encourage self-pollination. So cross-pollination is almost the same thing. You pick any two of these and try to run with it. It's basically this the opposite. So cross-pollination, if a given genotype is well suited for an environment, cross-pollination prevents this trait from being stable in the species. So if you have a genotype that's real good for that plant, well, it could be diluted or could be lost over a few generations, and it could, lose its stability in the environment. Again, this is unlikely to happen in most environments. Is usually again artificially run. You'd want to preserve certain traits. Cross-pollination promotes outbreeding, prevents inbreeding, which increases the amount of variation. So yeah, so cross-pollination, you have a lot of different plants that could be involved, subjected to different pressures, and you have a whole host and range of alleles that could spread through various offsprings. So yeah. 3, it increases vigor resistance. Again, this is just the opposite. 4, cross-pollination may introduce some undesirable characteristics, but new useful characteristics can also be introduced. So again, you want to have your bright, bright flowers, and suddenly you have a allele for an ugly flower in there. Well, it's undesirable, but it would increase genetic diversity. And as the last point here, it increases variation. Increased variation enhances adaptability to changing environments. So if you have a pest, or you have some kind of temperature change, or you have less water or more water, or whatever, at least some of the plants will be able to survive and will be able to proliferate and will be able to continue the species, and evolution would continue over time because you'd have a lot of variance for natural selection and evolutionary potential. And yeah, so that would eventually lead to speciation, which is one species branching out into many. So you pick, I would say, about 2 of these, and you run with, try to elaborate on it, and that is June 2019, and that should be it. Look forward to more. Laters.